Good afternoon and welcome to the IGIP seminar series presented to you by the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health. My name is Bene and I'll be your moderator for this talk. Today we're delighted to welcome Dr. Pia Abeltzourvish. Dr. Abeltzourvish is an associate professor of biology and early career chair of systems pharmacology in the Department of Biology at the Pennsylvania State University. Just a few standard logistics before we start. This seminar is being recorded, so please turn off your camera if you do not wish to be recorded. The recording will be available on YouTube and on our website at a later point in time. After approximately 45 minutes, we'll stop for questions. Throughout the talk, you can send questions via the chat function to James Hay. You have the possibility to ask your question to our presenter directly. In that case, please notify James in the chat. We will ask you to turn on your camera and we will unmute you once we're ready to take your question. Coming back to our speaker, Dr. Abeltzourvish grew up in Freiburg, Germany, where she attended Latin school, majoring in mathematics and chemistry. She went on to study biochemistry and molecular biology at the University of Kiel, Germany, Griffith University, Australia, and University of Basel, Switzerland. After getting her Master of Science with distinction, she worked at Roche Diagnostics and was an intern at the World Health Organization before beginning her PhD in theoretical biology at ETH Zurich, Switzerland, with Sebastian Bonhoeffer. Dr. Abeltzourvish received the ETH Medal for Outstanding PhD Thesis, which only 8% of PhD students get at ETH. And during her work as a research fellow with Ted Cohen at Harvard Medical School and Yale School of Public Health, she held several career development grants from the German Academic Exchange Service and the Swiss National Science Foundation. In 2015, she became a young associate investigator at the Norwegian Center for Molecular Medicine and joined the Department of Pharmacy at the Arctic University of Norway as a tenure-track associate professor. In 2020, she joined Pennsylvania State University as an associate professor of biology. And finally, for the last 2 months, she has been working with the Norwegian CDC on the COVID-19 response. Our speaker's talk is entitled At the Interface of Chemical Kinetics and Population Biology, How to Design Drug Dosing Strategies. Thank you very much for joining us. Dr. Take it away.
Yeah, hi, and thank you very much for the introduction. So you were able, I don't think I gave you all that detail, so I think you must have found it somewhere. So what I'm going to talk about today is mostly on how to design drug dosing strategies. But as Bene mentioned, there will be like currently I'm working with the Norwegian government and trying to help with the COVID pandemic. So there will be 2 parts. So one part where I'm talking about my main focus of research, and the other part where I'm talking about national models that are used in Norway. And I was asked to pick your brain with some of the things that we're struggling with. So I think that first, I'd like to talk a bit about the drug development process and why it's so expensive and takes so long. I think that now, since the pandemic started, people have been waiting for drugs against COVID. We don't really have good drugs yet, so that's a very good, reminder maybe how difficult it is to get effective drugs, whatever that the indication might be. And just to walk you briefly through the drug development process, so in the beginning, in drug discovery, you identify drugs that have activity in vitro. You then go on and test additionally with in vitro and with animal studies. Then in phase 1, you ask whether that drug is safe. During phase 2 and 3, you use increasing numbers of patients that actually have that disease and do dose ranging trials and ask whether the drug is actually effective. And then this is submitted, the material is submitted to regulatory bodies such as the FDA or EMA. And the question is very often, is that drug is at least as good as others? And that is not currently the question with COVID pandemic, but usually it is. And then after approval, you have these long-term observations in a very large number of patients. And typically that process takes decades. The costs of drug development have risen substantially, and that's mostly because of the clinical part. So the blue here, that's the costs for the preclinical part, and the red is the clinical part. So preclinical is everything until you go into animal models, and the clinical part are all the different trials in patients. And then you can see since the 1970s, the costs have really gone up, and now the clinical part, what's, recruiting patients and all these things, that's really the most expensive. One part of our, so the stuff that we focused on before COVID was TB, and we were especially focused on TB and trying to make the drug development process a bit smoother, because when you look at the health expenditure in some high burden TB countries, so for example, Afghanistan or Nigeria, you can see that it would be very difficult in terms of like investing into something to get your money back. And as you probably know, TB does not very often afflict people in Western countries. One way, so how do we try to help drug development? What we're doing is so-called PK/PD modeling. So if you think about how a drug acts in the body, so you have here, for example, a tablet, and inside that tablet are drug molecules. Those drug molecules are absorbed and get into the systemic circulation. And then at the site of action, the drug binds its target. You might dissociate again. And when the drug is bound to the target, you elicit some kind of drug response, and that makes you see that effect. Now the pharmacokinetic part, that's the part from you swallow that pill, and then it's somehow either, depending on where you measure, you look at how much ends up in the systemic circulation or at the site of action. And the pharmacodynamic part really starts when the drug binds its target and then all the way to the observed drug effect. That drug effect can be quite obscure. I mean, usually in infectious diseases, you measure pathogen load, but in other diseases, you could, like especially with neurological diseases, the drug effect that you observe can be quite complex. And then you couple those two models and come up with a so-called pharmacokinetic, pharmacodynamic model that allows you to simulate what would happen if you give a patient, let's say, a pill a day, or twice a day, or double the dose every 2 days, or something like that. The somehow a bit weird thing about this type of modeling, although it's like very widely used, and all pharmaceutical companies have big departments that focus on this, is that the pharmacodynamic models that are typically used in anti-infectives use an approach that was developed in 1910 by Hill. And that approach is not only like very old, I mean, it has its reasons to use it, but there are assumptions underlying this approach that are frequently violated. And these assumptions were made at that time such that you could basically, you don't need a computer to think about what would happen. This is different in many other diseases that give you more return per investment. So in for example, cancer or neurological diseases, people very often use more advanced pharmacodynamic models. So what does it mean, pharmacodynamics, in the context of antibiotics, or you can generalize that to anti-infectives? So here you have like a schematic drawing that should explain that. So this graph here is a very schematic drawing of the so-called bacterial time kill curve. Could be also any other pathogen, basically. So you have the timeline on the x-axis, and you have the logarithm of the pathogen or bacterial load on the y-axis. And then here in red, you observe pathogen growth without any addition of medication. And then you have an increasing concentration of drug. And the slope of that curve should become steeper and steeper. And then you translate that to a so-called pharmacodynamic curve, where you plot the net growth rate, basically the slope here, over the logarithm of the antibiotic concentration, and you get a typical sigmoidal curve. In the case of antibiotics, the point where it is exactly 0, so 0 net growth rate, would be the MIC. And a lot of the work that we've been doing is on relaxing the assumptions of these classical models. So basically, usually what you do is you somehow force bacterial behavior into a straight line, and then you make this curve and you fit a sigmoidal, just a sigmoidal curve through this, or hill function. And that is okay if you make certain assumptions. So these assumptions are that you're in equilibrium, that you have a constant concentration, and that basically the concentration outside of the bacteria equals the concentration inside of the bacteria. And now what happens when we relax these assumptions? So what if we use kinetics instead of equilibrium approaches? What happens if we allow for fluctuating concentrations? And that's very important, because there are very few examples where drug levels in a patient are constant. That happens, for example, when you lose beta-lactams, you sometimes in the ICU use constant infusion. But in the very vast majority of drugs, you always have something that goes up and down, like with a daily rhythm, for example. And then also, depending where the drug acts, the outside concentration outside of the bacterial cells is not necessarily the concentration on the target. So here is like how we envision, so what we model, basically a schematic of our model. So assume you have the antibiotic here in green inside of the cells, and then you have the target molecules in blue inside of the cells, and this weird black circle is the bacterium. Then first the drug has to get to the target, for example, by diffusion, and become intracellular antibiotic. Then it has to bind its target, and at a certain point, when enough target is bound, a threshold for killing or growth suppression is reached, and that would be your MIC. We're making here one very simple assumption, and that is that with increasing number of bound target molecules, replication decreases and or killing increases. And that is it's very, very hard to experimentally quantify. I'm not sure it would work if one did that, but it would be very hard to otherwise explain why increasing antibiotic dosages or drug doses in general would have an increasing effect on bacteria. So basically, if you want to like rephrase that, you have here, for example, you have a bacterium that has here these target molecules and here the antibiotic molecules. If there's relatively little target bound, they can still replicate. And the way we model this is that we assume that the bound target molecules are distributed over the two daughter cells by binomial sampling. And then you have, if you have a high number of bound target molecules here, then the cell dies and either releases its content into the outside medium or stays intact, depending on the antibiotic in question. If we simulate that, it looks like that. So here you have a simulated time kill curve. On the x-axis, you have the time in hours, and on the y-axis, you have the bacterial burden in CFU per milliliter. The black line is the cumulative, so the total bacterial count here. So at that, we would here be at an antibiotic concentration that is above the MIC, because the total bacterial number goes down with time. And then here the colors indicate how many target molecules would be bound. So you can see here blue is 0 bound target molecules, and that population disappears very, very quickly. And then as time proceeds, you have more and more bound target molecules, and with every bound target molecule, so this is a simulation for cephaloxacin, the cells are more likely to die and less likely to replicate, and that results in a total population decline. And we're using this model to try and optimize antibiotic therapy. So for example, we're asking like how often should a drug be taken? How much antibiotic should be given? How long should a patient be treated? And that is not really unique to antibiotics. People are fighting with exactly the same questions in all therapeutic areas. And it is very important to think about it a bit because these dose ranging trials and the question like, what do you first test in animals? You have an infinite amount of dosages that you could try. So it's really worth having an educated guess at the very beginning before you start testing the stuff in animals or in humans. And in antibiotics, it's actually very hard to answer those questions because, so to answer the question how often a patient should take drugs, we have to understand a bit better how bacterial growth remains suppressed even if the antibiotic is not physically outside the cells anymore. So that's called the post-antibiotic growth suppression. It's not easy either to understand how much antibiotic should be given because that depends on the bacterial density for some drugs, but not for others. And it's also not easy to understand how long a patient should be treated because that is complicated by persisting bacteria. It's a big problem in TB, for example, where it's really not that clear when you can safely stop drugs. Another thing that is something that pharmaceutical companies, for example, are very interested in is how should we quantify exposure? Like if we, like what I've shown you before, right? Those antibiotic pharmacodynamic curves, they assumed a constant dose, but this, as I said before, like the patient concentration of the patient is not constant. And typically what you see is here, so this is the time concentration profile of any kind of drug, basically. So you have here the timeline, and here you have the concentration. And then you have a typical time curve here, where in the first, right after ingestion of the drug, you don't see anything systematically. And then after a while, you reach the peak concentration, and that declines again. Now, for any kind of drug, what we need to do is to identify pharmacokinetic drivers. And those pharmacokinetic drivers could be the peak concentration, the area under the curve, or the time that the drug exceeds a certain threshold. And usually none of these is really good, or a mixture might be good. For example, when we are looking at TB, we have isoniazid as a mainstay of TB therapy. There, the peak concentration seems to drive the action. We have rifampicin, there the area under the curve is very important. And beta-lactams are emerging as a treatment option for TB, and we're working with clinical trials there. And there, the time above the threshold seems to be the best thing. So, and to understand these questions that I asked, like, how do we optimize dosing in terms of dosing frequency, dosing level, how long a patient should take drug, how do we optimally modulate the drug exposure profile to have maximal efficacy? So what we were able to show is that this schematic model that I've shown you, like just modeling how drugs bind in the bacteria and then kill the bacteria, can explain the post-antibiotic effect, can explain some of the persistence we see, and can explain dose response curves and density dependence. And importantly, independent groups later in the last years have verified a lot of the predictions that we made with these mathematical models. So now this is an overview of mostly current and some past members of my group who have worked on employing these models on different questions. So for example, Jingyi here is working on how to use parameters such as binding parameters that you can collect very early in drug development into optimal dosing. Antal was my PhD student, he just finished. He worked on predicting persistence in TB based on these molecular parameters. And Fabrizio is working on resistance. And then also we've also started working on combination therapy, and that's also driven by Fabrizio. We're using kind of a three-pronged approach where we are looking at, so that is, I think, what we published most on, because we first had to establish some of the methodology, like what happens, for example, if you have multi-step bacterial killing and not single-step bacterial killing. Then we also worked a bit on like proof of concept studies, where we validated our model with data from E. coli, because doing experiments TB is not very satisfying for the people doing the experiment. And then we are also currently working with them, and that's the main focus of our work currently. We are working with clinical TB trials, and then we're also trying to work more on COVID trials, where we're trying to apply those principles that we have the mathematical methodology. We were able to validate it in E. coli, and now we're trying to optimize treatment in drugs that are currently under development. So I wanted to show you briefly what we've done in E. coli. So basically, the assumptions that we made in this model is that somehow drug target binding shapes how bacteria react to antibiotics, right? And we had to we had to show that is actually the case. So basically, what we're trying to do here is to establish a link between chemical parameters such as drug target affinity or the intracellular drug concentration, or, for example, as one resistance mechanism might be that you get gene duplication or some downstream processes, with population parameters such as the MIC, or how much a bacterial population declines over time at a certain drug concentration. So I'll start with showing you some things that we published on predicting the MIC, because that is the simplest model. I mean, this is the part where one can write one equation. So so to come back to our schematic, one thing we need to know is like the gradient between the extracellular and not necessarily intracellular, but the target concentration, the concentration of the antibiotic at the target. We also need to know the binding affinity. We need to know ideally how much target a cell has. That is very often not the case. And we need to know the threshold. Luckily, we can do a lot by just assuming that you have some kind of percentage of threshold, and then you can forget about the amount of target molecules. So you just say, okay, 50% instead of 50 molecules, and you need to know how much target there was in the beginning, because that is really not that easy. And if we now interpret the MIC as the concentration, where at equilibrium, the target binding exactly is at the threshold for killing or growth suppression, you get a quite simple equation where the MIC depends on this concentration gradient, it depends on drug target affinity, and then it depends basically on the fraction of target fat. To put that back into like these chemical parameters that I've shown you, so you have the drug target affinity here, you have the intracellular drug concentration or efflux pumps or something like that here. You could model gene duplication, or basically in the simplest case, any kind of downstream process is just this critical threshold. And then we used, as an example, the antibiotic class of quinolones, which has been developed more and more over time. So initially, the first quinolone was nalidixic acid that had a very, 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 very poor KD. And then typically what people do if you have a drug that's okay, but not great, is that they try to improve target affinity. Now, that's usually a part in drug development that people work, like they see in the first drug. So now you would not market nalidixic acid anymore. Now you would immediately start, at least if you have an indication that brings enough money, you would immediately start to modify that until you get a good molecule. And then they modified that further. So oxalinic acid has a slightly better KD. So the affinity goes up when the KD goes down, I should say that. And ciprofloxacin in the end was really a much better binder to the target than the other previous quinolones. And then we grouped the quinolones by affinity. So we used from norfloxacin, which has probably the best target affinity to nalidixic acid. And we used historical collections of MICs before quinolones were rolled out in the clinic or widely rolled out. So you have drug target affinity here in the x-axis and the susceptibility of the MIC on the y-axis. And because all the affinity parameters were measured in Escherichia coli, we're just focusing here on Enterobacteriaceae because we assume that the targets are relatively conserved. So we collected data on 11 Enterobacteriaceae. And what you can see here is that in all the 11 species, you have a relatively good correlation between the drug target affinity that was measured in Escherichia coli and the MICs that were measured before quinolones were widely used in the clinic. So these are data before 1990. And at that time, quinolones were not that widely used. So ciprofloxacin was marketed later, and the others are not that good. And then we wanted to go from this very simplistic model to a realistic prediction. And then, like, how can we quantify this? Like, with each successive bound target molecule, replication decreases and or killing decreases. Like, how do we, it's nearly impossible. I think it's impossible to measure experimentally. I'm happy if someone can show me data that can do that. But basically, we're like, how do we move this to something that's really quantitative? So what we came up with then is that we wanted to infer that from bacterial responses to the drugs with a model. And the approach that we used was based on partial differential equations. So this is work that Fabrizio did. Fabrizio is a mathematician. I want to be able to handle a partial differential equation, probably. But basically, so what we previously had, just to explain you this graph. So this is the number of bacteria. And this is the timeline, like after the addition of drug that would happen at 0. And this is the percentage of bound target in the bacterial population. And what we previously did, so my approach that I developed in 2015 was to have as many OD, a system of ODEs with AE as with as many equations as there are target molecules. And that explodes quite quickly. So Fabrizio now had translated this, so this was published last year, he translated that into a PDE. And the nice thing about the PDE is that you can also very easily model combination therapy. Won't have time to talk about that today, but it's just, I think, much more elegant than what I did. And then we fitted the model to time kill curves. So this is E. coli exposed to ciprofloxacin. And we got a relatively good R-squared, if you believe in R-squared. And then we also inferred the MIC from this. So the MIC is not directly measured. And the MIC that we inferred was a range of what it was, the measured experimental MIC from the literature. We then wanted to show that we can change something about the way that the drug interacts with its target. So basically, the chemistry of or the biochemistry of the drug target interaction, and still be able to predict what happens. So what we were using is a set of strains that are overexpressing the target molecule of ciprofloxacin, that's the gyrase. That's the main target molecule. It has some side targets, but that's the most important target. And then we fitted our model to time kill curves with those overexpressed target molecules. So here you see basically that pharmacodynamic function. So you have the drug concentration, or logarithm of the drug concentration here on the x-axis, and you have the net growth rate on the y-axis, and then you have here in blue the wild type, and in red something which was mildly induced to overexpress gyrase, and in yellow something that was strongly induced to express gyrase. And then from the model fit, we wanted to infer how much is this overexpression, and then go back and quantify how much it was overexpressed. So what we did then is that we inferred from this like a dose response curve that's a bit different, where you don't have the drug concentration on the x-axis, but the amount of bound, or the percentage of bound targets relative to the wild type on the x-axis. And here I'm only showing the death rate. So it affects the growth rate too, but I'm only showing you the death rate here. And this here is the dose response curve, or the bound target response curve for the wild type. And then we assume that nothing changes in the other strains, just you know, when we still have a sigmoidal curve, because we always get sigmoidal curves when we do this. And from this fit, Fabrizio estimated that in this mild overexpression case, there would be 140% overexpression, and a strong overexpression case, there would be 220% overexpression. And then we used Western plots and quantified those Western plots, and quantified how much gyrase. So here are the two subunits of gyrase, and here is the control, how much overexpression we have found. And this is, so this is basically exactly what we predicted. So you have here, so the measured amount was, I think, 150, and here you have actually exactly 220%. So we were able to show that by fitting our model to time kill curves, we can predict the degree of a molecular change in a bacterium. And other people have in other diseases, I mean, this is a very evolving field, has come up in the last decade. So other people have made similar observations in other diseases. For example, in cancer and in neurological diseases. And what is a bit baffling to me is that drug target binding really predicts efficacy so well, because drug target binding is determined outside, like in an in vitro system, in an acellular system. And there's all kinds of theories saying that there should be a molecular crowding in cells that could change the binding parameters, for example. But still, this is like these novel approaches are so good that in those diseases where it's worth measuring, most major pharmaceutical companies are not saying anymore they do PK/PD modeling. They say they do PK/TE/PD modeling. So pharmacokinetics, target engagement, pharmacodynamics. So it seems to work, but I still find it a bit surprising that it works so well. And now it's a complete cut. So when I told my colleagues yesterday I'm going to give a talk here, then at the Norwegian equivalent of the CDC, they said, oh, we are having these problems, and can you please ask them? So now completely reversal. I'll tell you a bit what we're currently trying to do in Norway and what we don't really understand. So basically the part that I'm working on is pharmacological interventions in case vaccines don't work. I mean, we have several variants now around where vaccines are not that effective anymore. And we also have these settings, I think that in the US, there was the same, so that you have nursing homes where probably the are not without any interventions is so high that with the current vaccine efficacy, we can't suppress things. So we had now 3 nursing home outbreaks in Norway and fully vaccinated residents. So the basic idea that I had was like, what if we identify promising treatments early and look at the viral load, and then we try to implement that in we try to model that as national strategies. And then and the reason for that was that Norway has seen really strong vaccine side effects. Then we have these vaccine escape mutants and nursing home outbreaks. And one of the things that I noticed is that, it's not that well known how heterogeneous the side effect frequencies in European countries are. So Norway has been hit hardest by the side effects due to the AstraZeneca vaccine. And for that reason, our vaccination program is delayed and we need to look a bit at other strategies. So what we are seeing in Norway really frequently is something due to the AstraZeneca vaccine that's resembles the heparin-induced thrombosis, where antibodies are formed and also confirmed. So this is a recent publication in the New England Journal that came out from a Norwegian group. And all patients where this was seen, was confirmed that there are antibodies after the vaccination that recognize platelets and sequester platelets, thereby leading at the same time to a thrombosis, very often sinus vein thrombosis, and low platelet count and heavy bleeding at the same time. So we had people who died of intracranial bleeding in Norway. And what is really surprising when you look at the total numbers is that we have so much. So we had 5 confirmed cases with antibodies, one where we're not quite sure, so it could be 5 or 6 per 135 vaccinated people. And of those, 3 are dead. And this level of side effects is very much higher than in any other European country, and you see a lot of heterogeneity. In Denmark, in the side effect frequency, so we are between 1 in 20 to 1 in 30,000 in terms of side effects, really serious side effects. In other countries, such as Denmark, it's 1 in 40,000. In Germany, it's between 1 in 70 to 1 in 150,000. And these might be the numbers that you're most familiar with in the UK, just because so many people were vaccinated in the UK. These numbers dominated when you look at the total European area where AstraZeneca has been approved. And we're really struggling to understand why, whether it's a weird Viking gene that makes people get side effects in Norway, because this has been really well shown, because this has been molecularly analyzed. And then just yesterday, there came a preprint out from my colleagues where they were able to show that men get 1% mild bleeding after the AstraZeneca vaccine. So like gum bleeding after brushing the teeth, nose bleeding, like little red skin dots, and women 3%. And this doesn't happen in the mRNA vaccines. So I don't know what that means. No one does that. And it could be like the composition of the vaccinated population, or it could be true genetic reasons. And it would be hard to find out, I guess, why. But what kind of genetics would be interesting to find out whether it's a real difference. So we're working on that, because if it's a real biological difference, then people in Norway might also be more susceptible to side effects due to the Johnson & Johnson vaccine. And then we should not roll it out. And so that is because when you look at the outcomes of COVID infections in Norway, so this is basically in the first, like in last year. And you look at death, ICU, or hospitalization as an outcome in different age groups, so from 0 to 19 to 80 plus, and you compare that to the side effect frequency that were mostly healthcare workers were vaccinated in Norway, so 20 to 39 and 40 to 59. If you compare that to the serious side effect frequency, so ICU or death, you see that we're in some age groups, even getting the vaccine is much worse than getting COVID. So, because of this so basically what the Norwegian government has now done is AstraZeneca is on pause, and they are having, because we don't really know why our numbers are so different, they have formed an outside expert committee to evaluate whether AstraZeneca should be made, be given outside of the usual vaccine scheduling, just for volunteers who can choose to be more quickly vaccinated. Maybe men, for example, because we see more bleeding in women, if they want to. But I don't think it will come back, to be honest, because it's very, very hard to defend, especially because we have very, very low COVID frequencies, and it's very hard to defend, to tell like someone in this age group that you're going to vaccinate them when they are much more likely to die from that than from COVID itself. So so it's very doubtful that we will get vector vaccines, I think, with a bit too many safety concerns. And as I said, it would be important to understand this because Johnson & Johnson seems to have similar problems, also at a very low frequency, but maybe the frequency in Norway would be also much higher than in other countries. So we don't know yet. The other project that we're currently working on is so one of the vaccines can't be used in Norway. So, and then we also have, as you all know, we have all the variants around. And what we're currently trying to do is simulate a worst case scenario, or that worst case, I mean, it's maybe not that unrealistic that, for example, the Brazilian P1 variant arrives in Norway and starts to spread. So this is just for I'm sure that a lot of you are very familiar with that, but for those who are not, so the P1 variant was detected in the city of Manaus that had a huge problem last spring with a really, really big outbreak, and then had a very high seroprevalence. I believe it was around 76%. And then, so this was the first outbreak. And then after they thought they had reached herd immunity, they got a much worse outbreak afterwards. And there was a science paper last week that said that the new variant is roughly 2 times more transmissible. And also, as you can see from this, even though you have 76% seroprevalence, you still get an outbreak. It's probably not going to work well with the vaccines. So we have a national, so Norway has a very, very good control, knows a lot about its population. And from Germany, I find it sometimes a bit creepy, but Norway collects a lot of data, and there's a high trust in government, so people do share those data. We have a national model that models all the different regions in Norway. We have the age structure and comorbidities from a national registry. We have cell phone data. We have very good contact tracing. So locally, I think that just 1 out of 80 cases, when we have cases, is unknown source. And in other areas, it's around 25% unknown. The borders are closed with quarantine hotels. So basically, we wanted to model the an invasion of, for example, the P1 variant. We wanted to look at disease severity depending on age and comorbidities, and then and also vaccine efficacies, but that's not the part that I'm working on, and then model how we could contain that if that happens. I found that, and probably a lot of you know more about that me, that in the age groups between 20 and 60, you have a strongly increase, you have a strong increase in case fatality ratios, so between 2 and 3-fold, and hospitalization.
I think our presenter froze. So our apologies about that. We'll try and see if she can come back in the next second. Yeah, she left the call, so I guess we'll wait to see if she's able to come back.
I'm sorry. Somehow the connection was not as good. So to come back, I am very sorry about this. I am looking forward to the time where the whole internet stuff is not necessary any longer. Anyway, so these are the data that we currently don't understand. So we came across the recommendation of the Brazilian Ministry of Health that all women should delay pregnancy right now. So we went into the databases of the Brazilian Ministry of Health and looked at case fatality ratios in pregnant women, and they're really, really high. So they go up here in this province, it's called Roraima, they go up to 80%, which I find really weird. And the totals, when you look at the total picture in Roraima, both at what totally was reported and what is reported since the last update, it doesn't seem to be that unusual. So it doesn't seem to be the case that only pregnant women are, that only people are diagnosed with COVID who are going to die. This is an area where they have a lot of indigenous populations. So it has been reported previously that they have a really high mortality rate of COVID. And then I looked at that a bit further and looked at the age structure and disease severity in pregnant women in 2020 in total, so all of Brazil and the Northeast, and also in 2021. And in both cases, this has doubled. And now the question is like what we should do with this, because I was not able to confirm that in any way, but we're like, we have to update the national vaccination strategy by May 11. And the question is, what do we do with this? And if we can, because if we can confirm it, then we would change our vaccination strategy to do ring vaccinations around pregnant women, just in case this variant comes to Norway at some point. But we really, there's no, it's extremely hard to find any data on this. We were not able to establish a solid contact with people in Brazil. And if any one of you knows more about what's going on there, we would be super grateful. Because this is concerning, but we don't know whether it's true. And then the other thing is that there were also relatively vague reports that there is a really high mortality rate in infants. There was even an investigative report in BBC, but I haven't managed to find anything solid about that. So just like vague references. And we are interested in knowing this so we can recommend like how tight the borders should be and how long to keep travelers in quarantine, and whether to test them immediately for this variant and all these things. So the question is basically, is this real? Is it like really this PV1 variant, or is it something that's further mutated? Should we be really super careful around pregnant women, potentially also children below 1? We don't know. And so when we have all the parameters ready, what we will do is we will use the national model with and without vector vaccine rollout, probably without. and parameterize it with age and pregnancy specific outcomes, and then evaluate stricter quarantine or pharmacological measures on a national scale. So far, the model can predict, I think, 3 weeks into the future. So on the national scale, let's see what it would do when we start changing the parameters. I don't know. We'll see. Yeah, so sorry for the abrupt transition in the team. So I'm currently not doing what I'm usually doing. Yeah, and I'm happy to take any questions you might have. And if you have any idea what is going on in Brazil or why Norway, the Norwegians are dying of the AstraZeneca vaccine, we'd be very grateful.
Thank you very much for your terrific presentation, Dr. Abelstrovic. We have actually a few questions, and since COVID is the topic that you seem to be willing to prioritize, we will start with this, and then we'll move on to more to questions on the model you presented before. So the first question is from Leiden James. Regarding the differing rates of the sinus vein thrombosis following the AstraZeneca vaccine, is it in any way possible that the true probability is the same for all the countries, but due to stochastic uncertainty, we're seeing different small numbers of cases in these countries?
I think so. When my colleagues looked at that, they found it highly unlikely because there are orders of magnitude in between. But we'll have to look at that further, knowing more exactly about. So the problem right now, I think we have a pretty good overview over the age structure. What is a bit more complicated is that there might be an estrogen link as well. So we would have to, and I don't think that all countries necessarily know whether the women who were vaccinated were using contraception or not.
Okay. I think Professor Mark Lipsitch had a few questions for you. So we will unmute you. Unmute him, sorry.
Hi, Pia. Nice to see you. Just one. I wish I had anything useful to say about all these interesting questions you raised at the end, but I don't know what to say. I guess one question is it clear that the deaths in pregnancy are at high rates are from P1? Because my sense was that nobody knows what's causing like the frequency in different states in Brazil.
So apart from this weird press release by the Brazilian government, there were lower mortality rates throughout 2020 and higher mortalities now in 2021 that the strain has spread. There are also lower mortality rates in the south, which has so far had less P1 than in the northeast. It seems to peak in the northeast. And the government specifically said it is because of these variants that we're recommending not to become pregnant right now. I wish I knew. Yeah. I have been struggling with my very rusty Latin. I've been struggling with particular databases in Portuguese. I wish I knew better, but I don't.
The person at our school who I checked, I don't think is on this seminar, but who's Brazilian and has been working a lot on COVID is Marcia Castro.
Okay.
So it should be worth getting in touch with.
Yeah.
So I had I'm not sure if it's an interesting question or a technicality about your MIC model. I'm wondering how you get to the idea that at equilibrium, the average bacterium is right at the death threshold, because I would have thought that would mean much more than zero growth. It would be like really fast killing if the average bacterium was.
Yeah, it really depends on how you define that threshold. I should have explained that better. So we're explaining that threshold by, it's just not viable anymore. Just not viable anymore.
I see. I see. So it's a little bit arbitrary, I guess, where you put the cutoff.
It is, and this is not quantitative at all. It's about understanding which factors might play a role. It is useful if you, for example, think about further developing antibiotics. So what kind of changes in MIC would you get if you were able to make a new antibiotic that has this much more affinity? But I think that the quantitative approach is much more useful.
Got it. Okay, thanks.
Thank you.
Thank you, Professor Lipchitz and Dr. Abelsovich. The next question is from Dr. James Hay, my co-host, so maybe we can also unmute him.
Hey, is that working? Hey, thanks. That was like very comprehensive and thorough talk. You covered a lot of ground. I have one kind of very speculative kind of comment on the situation in Brazil, and then one also quite speculative question to you about dosing regulations. So the kind of point about the increased adverse COVID-associated complications in pregnant women in Brazil. It's just weirdly coincidental to me that there was a similar kind of phenomenon during Zika, where in that northeast Brazil there was far higher rates of congenital Zika syndrome per pregnant woman in that same region. It was Bahia, so not Roraima, if I recall. But it feels like a bit of a tinfoil conspiracy theory. But I wonder if there's something in that coincidence, about that population that is kind of predisposing pregnant women or fetuses to disease. So I don't know, maybe something to speculate on. The other question, again, very speculative, but it's about the regulatory framework for drugs and vaccines and diagnostics for a kind of an intervention that has a public health use rather than a clinical use. Do you feel that the regulatory framework really takes into account things like dose sparing and like kind of population level benefit as opposed to like an individual level clinical profile, right? So it might be that it's better to kind of compromise on dose in favor of having more vaccine doses, for example, even though the kind of per dose efficacy might be lower, so the population benefit might be more optimized.
I think it probably is very country dependent. I know that so in Norway, for example, there's not the distinction between different parts of governmental agencies like FDA and CDC in the US. It's like more like one. So, and the part that I'm doing now is to evaluate the population level efficacy for pharmacological interventions, and also the willingness to pay. I think, like how much they are willing to invest. So basically, the baseline here is you can't you can't force a patient to sacrifice themselves for others. So that's the reason why AstraZeneca is on hold right now, even though it might benefit older age groups. But like whether things should be invested into and so on, that's also evaluated on a population level. But I think it's also a bit mentality dependent. So I think that probably Germany would would find the German government would find the population level benefits less important than the Norwegian one, because it's a bit different perspective on like how to decide on things. I'm not that familiar with other regulatory frameworks, so I don't know how it is in other countries.
No, I think it's just an interesting thought from your talk, because we've been thinking about it in the context of regulatory approval for diagnostic tests, where the FDA is kind of wouldn't want to approve a rapid test because that's a lower sensitivity compared to like a gold standard. But then that part of that's the first hurdle to becoming approved, but then you can't get to actually providing a population level benefit. Like, that benefit's not a part of the regulatory decision. And I wonder if there's like parallels in pharmaceuticals as well. I mean, I suppose antivirals aren't really used typically as kind of transmission reducing interventions, but certainly vaccines are. And I guess you could imagine maybe an antiviral might have that like possibility.
Yeah, so currently we are mostly, well, I mean, to be fair, Norway doesn't really have so much of a problem. So we've been getting away with quite a mild interventions. But so far we're mostly evaluating vaccines, and then also thinking a bit about test and treat strategies in nursing homes. Because I feel that people there are becoming quite exhausted with all the measures that they have to follow. But the problem is we don't really have good data on reduction in viral load yet. So remdesivir, for example, doesn't do that much, unfortunately, maybe a little. Then the Regeneron antibody seems to reduce viral load, but that has to be an infusion. That is a barrier. I think there are a couple of drugs in the pipeline that look quite promising. And I don't know whether you've seen that there is something that was originally used as an asthma spray that can alter the clinical course, but I'm not sure because it's a steroid. I would suspect it works mostly on the host and not so much on the virus. But being able to do more test and treat, or reducing the infectious period would be super attractive. And that should also, in my opinion, it should inform how much governments are willing to pay in these national programs for an antiviral.
Interesting, thank you.
Thank you, James. And I think in the interest of time, we should stop here. On behalf of the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health, let's thank Dr. Abel Survish again for joining us today for her talk and the insightful discussion that followed. Thank you as well to everyone in the audience for your engagement and for staying with us despite the technical difficulties. Please do get in touch with any of the seminar organizers if you have any questions for us or our speaker. Please check out the CCDD website to stay up to date on upcoming seminars and other events. Thank you everyone for joining and enjoy the rest of your day.
Thanks.